relationship between viruses in North Africa. The floor is yours.
Good morning, everybody. I have to say that this work is a collaborative study between ISLER, that is my institute, and ANSES Institute from France, and with the colleagues from Algeria and Tunisia. From April 2014 to November 2015, FMD outbreaks occurred in the Maghreb region. The Maghreb is located in North Africa, and it constituted by 3 different countries: Tunisia, Algeria, and Morocco. This was the first FMD incursion after 15 years of absence, and the phylogenetic analysis based on VP1 region revealed that these viruses belonged to the type O in the 2001 lineage, within the Middle East South Asia topotype. This lineage was first identified in Indian subcontinent in 2001, and from that period it became predominant in India subcontinent, with sporadic outbreaks in Arabia Peninsula, and in 2013 it was detected for the first time in North Africa, in Libya here. So in order to get inside the evolution of these viruses and the epidemiological relationship between the outbreak during the epidemic wave in Maghreb, we collected 12 isolates from Tunisia and 5 isolates from Algeria all along the epidemic wave, and we performed the phylogenetic analysis. We studied the antigenic profile of some of the isolates. For the phylogenetic analysis, we prepared the samples, and so we submitted the samples to the MySeq instrument from Illumina in order to obtain a full genome sequence. And in fact, we obtained an almost complete full genome sequence of 8,000 nucleotides. And regarding the antigenic profile, we study the antigenic profile using a panel of monoclonal antibodies, of 55 monoclonal antibodies that are divided in 22 monoclonal antibodies recognizing neutralizing epitopes, and 23 monoclonal antibodies recognizing non-neutralizing epitopes. At first, we perform a Bayesian phylogenetic analysis based on VP1 gene. This enabled us to add more sequence from Tunisia and Algeria, for which wasn't available the full genome sequence. And it also, we had other viruses from Libya and Morocco, and that are highlighted with different color code. This analysis shown that a unique introduction occurred in North Africa, here, in July 2013, since the presence of a unique common ancestor from which all the viruses detected in North Africa have evolved. From this ancestor generate a cluster of viruses that evolved in the second half of 2013 in Libya, and then from Libya in Maghreb. All the viruses evolved in Maghreb originated from a unique common ancestor. This means that a unique introduction occurred from Libya to Maghreb. In Maghreb, the viruses evolved in Tunisia, and the tree support for two different introductions in Algeria. In fact, Algerian isolates from 2014 clusterized with other Tunisian isolates from the same period and sharing a common putative ancestor with high estimate posterior. On contrary, in Algerian isolates from 2015 are related to other Tunisian isolates and share a common ancestor with the Moroccan viruses isolated in 2015. We performed also a phylogenetic analysis based on the whole polyprotein codifying region. With this analysis confirmed that, that a unique common ancestor was present between all the Algerian isolates of 2014 and the Tunisian isolates. And also that Moroccan isolate was confirmed closely related with the second introduction in Algeria in 2015. And with this analysis, we evidenced that in Tunisia, the viruses evolved in 3 main branches. The first one is this one, in 3 main branches that were localized in 3 different geographical overlapping regions that I'm going to show you. This is the first one that include isolates from the provinces from the south to the north of the Tunisia, related to the North Algerian isolates. The second one include isolates in the central part of the Tunisia, and the third one in the north of the country. The isolates from Algeria in 2015 is localized geographically apart from the others. We also perform on the whole polyprotein codifying region another phylogenetic analysis, a statistical parsimony network. In this case, dotted lines represent genetic intermediates that are separated by one nucleotide change. The stars represents non-synonymous substitution. Red stars non-synonymous substitution in structural protein, and the yellow one non-synonymous substitution in non-structural proteins. We analyzed all the Tunisian isolates and Algerian isolates for 2014. And with this analysis, a common ancestor was confirmed between all these isolates, and also we detected a total of 48 non-synonymous substitution. 12 of these substitution involved structural protein, and some of them targeted different neutralizing sites. Also, with the analysis, we highlight a high number of nucleotide substitution between and within clusters. We perform also, as I told you, an antigenic profile by monoclonal antibodies. You can see in these 2 tables, we divided the non-neutralizing MABs in the right table and neutralizing MABs in the left table. All the isolates that we tested are chronologically ordered above each tables, and are also listed the prototype viruses against which the monoclonal antibodies were generated. The different cells represent a different reactivity of each MABs against the isolates. In the table on the left, we listed all the monoclonal antibodies, neutralizing monoclonal antibodies that are with different color code in referring to a different target size, neutralizing target size if it is known. Regarding, you can see that the prototype viruses show a distinct profile and 3 different profiles with the neutralizing MABs. Instead, all the North Africa isolate show a homogeneous profile, with the exception of some MABs that may be recognized not stable epitopes. And also there is this strain that has a different profile and maybe has to be studied. With non-neutralizing MAB, the situation is even more homogeneous. The reactivity is even more homogeneous, considering also that These epitopes are not submitted to a selective pressure like neutralizing MABs, neutralizing epitopes. So in conclusion, all Tunisian isolates shared a common putative ancestor and evolved in different branches in Tunisia, probably in relation, but we have to study more inside with animal movements. Two different FMDV introduction occurred in Algeria. Many nucleotide substitutions were detected between and within isolates, and probable undetected FMDV infection, or we don't have enough, analyzed enough samples. And there was also a limited antigenic variation among isolates restricted to neutralizing MABs. I have finished. Thank you for your attention.
Thank you very much, Julia. It's fascinating. Any questions from the floor? Maybe while people are thinking, I was interested in the data that you presented for the differences between the different monoclonals. So you have viruses that are genetically very closely related to each other, but you have a little bit different profiles, I think, if I understand. So how do you do those experiments, and how do you ensure that the differences that you see are due to the viruses rather than maybe the growth of the virus in the cell or the amount of virus that is tested? Do you understand the question?
No, I don't understand.
So how do you, for the monoclonal experiments, how do you show that the changes that you see are not due to differences in the amount of virus that you've added into the study? I mean, how do you control for this study?
Yes, maybe I can answer the question. The ELISA with monoclonal antibodies is a standardized ELISA, and we try to use the same amount of viruses. We have a titration of viruses in a polyclonal ELISA, and then we do we try to control that the amount of virus is the same, and we try to use very initial passages on cell culture. But of course, we cannot exclude that some variation may occur during cell cultures. And we don't think the variation Julia showed are important variation.
Okay. Are there any other questions? I think Keith has another question, maybe, at the front here.
Thank you. I think all of the viruses you looked at were from materials collected from acute cases during outbreaks. But there was also some serology undertaken in some of the countries. So presumably, probang samples collection from seropositive animals might give you some of the missing links, or could have picked up some of those subclinical infections a bit earlier. Would you agree?
Yes, we have to ask our colleague in Tunisia if they collect or provided us more samples in order to get inside what we detected at first.
Okay, thank you very much, Julia.
Okay, so we move to the next speaker, but